Hello, uh, we want to learn together the mechanisms of plant signal transduction. I read these two pages, but unfortunately the video doesn't have voice. We learn together about the plant defense against abiotic and biotic stresses, heat, cold, drought, salt, metals, flooding, and also about the phytohormone pathogen mechanism, pathogen attacks and insect attacks, biotic stresses, plant development progress through distinct phases, vegetative growth, followed by reproductive phases. Seeding establishment is a vulnerable stage. About the different enzyme these phases attack during the vegetative phase, generally rapidly increased by their photosynthetic capacity and their size and mass. Now we reach here. Secreted cysteine-rich peptides represent one of the main classes of signaling peptides in plant, whereas the post-transitionally modified small non-CRP peptides are mostly involved in signaling event during vegetative development and interaction with the environment. Cysteine-rich peptides, CRP, are overrepresented in reproductive processes including pollen germination and growth, self-incompatibility, gamete activation and fusion, as well as seed development. Tip growth of the pollen tube depends on oscillatory exocytosis focusing at the apex, which is regulated by a RAC, ROP, or row type small GTPase in plants. Rows and cytosolic calcium ion concentration at the tip to form a fine tubular structure, exocytosis and endocytosis, as well as mechanical properties of the cell wall have to be monitored and precisely controlled. In the last decade, emerging evidence has suggested that receptor-like kinase or RLK, serine kinase with a single transmembrane domain, work at the pollen tube tip to transduce external signal into the tube. Currently, many but not all key CRP ligands and RLK are available. Leaf cell imaging and structural analysis uh, of the dynamic of ligand receptor complexes as well as downstream signal transduction will be important direction of future research. Seed germination is another complex process influenced by many physical factors such as water, temperature, and light, by a great number of chemical factors such as abscisic and gibberellic acid, reactive oxygen and nitrogen species that play vital roles in regulation of dormancy. Seed germination is initiated by water imbibition at appropriate temperature. During germination, nitric oxide content and the level of protein nitrosylation increase, exerting a major influence on seed germination and various sulfur nitrosylation protein control. Many kinds of redox-based regulation, such as reaction between protein thiols and thiol disulfide exchange, although there are morphological differences between mono cotyledonous and dicotyledonous seeds. The fundamental regulation of seed germination is similar in both groups of plants. Also, sugar and auxin may be simple molecules. They have Although sugar and auxin may be simple molecules, they have long been seen as signal in vascular tissue differentiation. Furthermore, other complex signal transduction circuitry must be present to ensure the stability of morphogenesis. The overall form of levels root, and flowers in chaotic environmental condition, although some plant characteristics are resistant to environmental variation. Many others respond plastically adding an extra layer of morphogenetic and biochemical complexity that is large unique to plants. Cytokine, cytokines, opsin, opsins, ethylene, and gibberellin, as well as the agents that help to strengthen plasticity. Ethylene, gaseous plant hormone, for instance, is an olefin hydrocarbon produced by all plants. Despite its simple chemical structure, its orchestra a myriad of complex functions. Controls processes as diverse as germination, root hair development, root nodulation, senses of organ, including fruit ripening, differential cell growth, absorption stress responses, and resistance to necrotrophic pathogens. It is known that ethylene is effective to include biological responses at a nanomolar concentration and that is responses only take minutes to be induced. Role of various hormones in plant life cycle. Okay, now we have a three hormone here, gibberellin, auxin, and mm, steroid. We have a vegetative growth here, flowering, reproductive growth, leaf serration, internode elongation, reproduction, embryogenesis, embryo. Then we have a seed, germination, hypocotyl elongation, and root growth, seeding again. Lateral organ formation. Okay, gibberellin is a blue. Gibberellin has a role in the lateral organ formation, lateral organ, in the leaf serration, in the internode elongation, in the reproduction, and in the germination, and in the hypocotyl elongation. Auxin has role also again in the lateral organ formation, in the leaf serration, in internode elongation, in reproduction, in embryogenesis, in hypocotyl elongation, and in the root growth. Brazinosteroid has a role in the reproduction, in the embryogenesis, the germination, and in the hypocotyl elongation, and in the root growth. This is the role of various hormones. Facile screens have been developed that precisely identify mutant plants that are either responses deficient and fail to respond to a stimulus, or response constitutive and respond in the absence of the signal. Genetic analyses have been performed to elucidate both the order and interaction of pathway components and suggest that a complex network of information input and interaction components are transmitted to the nucleus to direct change in gene expression. For example, Arabidopsis mutants that are insensitive to a phytohormone, gibberellin, GA, or those defective in its biosynthesis show a delayed appearance of the first adult leaf. Inter, interest, interest, Interestingly, another class of mutants that display accelerated vegetative phase changes are those associated with mutation in genes involved in the biogenesis and action of microRNA and of trans-acting small interfering RNA (TASI) whereas the effects of uh, interfering RNA or trans-acting RNA, TASI RNAs, on heteroblasty may be limited to their ability to affect leaf polarity via their targeting of auxin response factors. The role of microRNAs is more diverse. For example, microR 156 promotes the juvenile phase and when constitutively expresses cause a moderate delay in flowering. Regarding reaction of plants to biotic factors, especially fungal disease, plant resistance can be adult, which provides protection in a crop post seedling stages can be as early as tillering or late. What is interesting, cooler seasons delay the resistance the trait being expressed. And seedling, which by comparison is effective at all growth stages. APR is provided by one or more minor genes, which have only minor effects and on their own may not protect the plant from rust attacks. In combination of 2 to 3 effect is cumulative and the variety can consequently show high level of adult plant resistance, whereas seedling genes are called major and have larger effects, being constantly under attack by pathogens. Plants have evolved sophisticated immune systems to combat pathogen attacks. Upon the pathogen attack, plasma membrane localized pattern recognition receptor PRPS, including receptor like kinase or receptor like protein, recognizes pathogen associated molecular pattern to activate ramp trigger immunity PTI. In response, some pathogens can deliver effector protein into host cell to suppress the PTI. As a countermeasure, the pathogen effector can be recognized by the intracellular nucleotide binding and leucine rich receptor NLR. Immunoreceptor receptors either directly or indirectly, which in turn can activate effector triggered immunity. Both PTI and ETI share significant overlapping downstream defense signaling events, but ETI is much faster acting and mounts a more effective responses than PTI. The downstream signal transduction events may include NADPH oxidase dependent reactive oxygen species production, activation of calcium, mitogen activated protein kinase, and trimeric G protein signaling pathway, induction of salicylic acid biosynthesis, pathogenesis related genes, callus deposition, and biosynthesis of antimicrobial phytoalexins, as well as transcriptome reprogramming. The defense signaling pathway are interconnected to form a defense network in which numerous transcription factor biochemical pathway and phytohormones are involved. Interestingly, some resemblance between the hypersensitive response and animal programmed cell death were noted. Leaf synthesis is the cumulative response of multiple factors operating at the final stages of the plant life cycle. It is the generative process that is genetically controlled and involves nutrient remobilization prior to death of a leaf tissue. Age is a key development determinant of the process along with other synthesis sen- senses senses inducing factor. Preferential breakdown of chlorophyll leads to the color changes of monocarpic plants from green to golden yellow as the grain from as the grain matures before harvesting. Furthermore, chlorophyll breakdown in combination with synthesis of new pigment like anthocyanin and phenols due to hydrolysis of micromolecules give a conspicuous range of colors in the autumn leaf of deciduous trees. Deciduous trees. At the cellular level, different hormone signaling molecules and transcription factors contribute to a regulation of synthesis. For instance, Arabidopsis mutant deficient in GA biosynthesis or GA signaling displayed delayed synthesis phenotypes supporting the role of GA as a negative player in the regulating leaf synthesis. Genes that are upregulated during synthesis are generally termed as a synthesis associated gene, while genes or downregulated expression patterns such as photosynthetic gene are termed as a synthesis downregulated gene. Transcription factors as a regulatory protein serving as a switched in the processes of different gene expression have been shown to be involved in both the promotion and or inhibition of plant synthesis, senescence, 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 or such like this. Sorry for my pronunciation. Based on DNA binding domains among the 2403 TF encoding gene on the Arabidopsis genome, at least 287 transcription factors in 34 gene families have been shown to be synthesis associated, including membrane in the NAC, WRKY, MYB, C2H2, APE2, and HB gene families. As the environmental context changes, so will the strengths of connections vary between the protein and other molecules that make up the networks. A strong spatial dimension underlies signal transduction. For example, plants must be able to delete gradient in signals such as light and resources such as nitrate and water. Plant development itself also is uh, decidedly polar. The spatial dimension is satisfied in many ways. Cell place receptors, channels G protein, and kinase is a specific membrane. Some signaling protein complexes are permanent, such as the relative stable and perhaps hardwired constitutive photomorphogenesis signalosome COP9. Other signaling protein complexes are likely to be ephemeral and formed immediately as a result of signal. There are at least 600 receptor kinases in Arabidopsis, and most of them are membrane bound incompatibility and disease defense signal transduction. Use receptor kinase after ligand binding and autophosphorylation. Such kinase may act as nucleation site for the construction of ephemeral signaling complexes that contain MEI protein. Many other GTPase 1433 protein kinases and phosphatase may cluster around these nucleation sites, forming kinase cascades and the regulation of specific transcription factors. For example, by phosphorylation will complete the pathway. Okay, thank you so much from the Professor AZ from the Biotech and Polytech University for this note.